Alchemical Wizard 18, Epigenetic Health/helping our dormant DNA reignite. Epigenetics is the study of heritable changes in gene expression, active versus inactive genes, that do not involve changes to the underlying DNA sequence, a change in phenotype without a change in genotype, which in turn affects how cells read the genes. Epigenetic change is a regular and natural occurrence but can also be influenced by several factors including age, the environment/lifestyle, and disease state. Epigenetic modifications can manifest as commonly as the manner in which cells terminally differentiate to end up as skin cells, liver cells, brain cells, etc. Or, epigenetic change can have more damaging effects that can result in diseases like cancer. At least 3 systems including DNA methylation, histone modification, and non-coding RNA-associated gene silencing are currently considered to initiate and sustain epigenetic change. The nutrients we extract from food enter metabolic pathways where they are manipulated, modified, and molded into molecules the body can use. One such pathway is responsible for making methyl groups, important epigenetic tags that silence genes. Familiar nutrients like folic acid, B vitamins, and SAMe, S-adenosylmethionine, a popular over-the-counter supplement, are key components of this methyl-making pathway. Diets high in these methyl-donating nutrients can rapidly alter gene expression, especially during early development when the epigenome is first being established. DNA methylation is a major epigenetic modification involving the addition of a methyl group to the 5' position of cytosine by DNA methyltransferase to form 5-methylcytosine, 5-MC. This epigenetic mark has the power to turn genes on or off and can be inherited through cell division. Diet has also been shown to modify epigenetic tags in significant ways. The field of nutriepigenomics explores how food and epigenetics work together to influence health and well-being. Okay. So what other foods, vitamins, and supplements can we take to help us methylate and perhaps reignite our dormant deoxyribonucleic acid, DNA, in this ascension process? 1-methionine. Sesame seeds, Brazil nuts, fish, peppers, spinach. Epigenetic role, SAM synthesis. 2. Folic acid. Leafy green vegetables, sunflower seeds, nutritional yeast, liver. Epigenetic role, methionine synthesis. 3. Vitamin B12. Meat, liver, shellfish, milk. Epigenetic role, methionine synthesis. 4. Vitamin B6. Meats, whole grains, vegetables, nuts. Epigenetic role, methionine synthesis. 5. SAMe, SAM. Supplement. Epigenetic role, enzymes transfer methyl groups from SAM directly to the DNA. 6. Choline. Egg yolks, liver, soy, cooked beef, chicken, veal, and turkey. Epigenetic role, methyl donor to SAM. 7. Betaine. Wheat, spinach, shellfish, and sugar beets. Epigenetic role, break down the toxic byproducts of SAM synthesis. 8. Genistein. Soy, soy products. Epigenetic role, increased methylation. 9. Sulforaphane. Broccoli. Epigenetic role, increased histone acetylation turning on anti-cancer genes. 10. Butyrate. Fermented foods. Epigenetic role, increased histone acetylation turning on protective genes, increased lifespan. 11. Diallyl sulfide, DADS. Garlic. Epigenetic role, increased histone acetylation turning on anti-cancer genes. 12. Curcumin. Turmeric. Epigenetic role, MICR orna, myrna, histone modifications, DNA methylation. 13. Vitamin C, L-ascorbic acid, is an essential, water-soluble micronutrient that exists predominantly as the ascorbate anion under physiological pH conditions. It is well established that ascorbate is an antioxidant and free radical scavenger and further, an essential cofactor in numerous enzymatic reactions. Guavas, papayas, oranges, kale, broccoli, kiwi fruit, strawberries, acerola cherries, rose hips, chili peppers, thyme, parsley, lemons. Epigenetic role: ascorbate serves as a cofactor for 10-11 translocation, TET, dioxygenases that catalyze the oxidation of 5-methylcytosine, 5-MC, into 5-hydroxymethylcytosine, 5-HMC, further to 5-formylcytosine, 5-FC, and 5-carboxylcytosine, 5-CAC, which are ultimately replaced by unmodified cytosine. The JMJC domain containing histone demethylases also require ascorbate as a cofactor for histone demethylation. Thus, by primarily participating in the demethylation of both DNA and histones, ascorbate appears to be a mediator of the interface between the genome and environment. Perhaps this will inspire us to eat healthier and to consider a diet high in methyl-donating nutrients. All of our epigenomes are different. Our blood types are different. We all can agree that vegetables and fruits play a key role in our metabolisms and epigenetic processes. Stay hydrated, my friends. We are sovereign, we are free. Travis Carper. January 22, 2019. Blog on 3decrees.org.